Now we will start for next bacterial disease in medical microbiology paper. Now this disease is known as Staphylococcus infections. Now this bacteria is involved in various kinds of diseases which occurs in human beings. And these infections, they are mostly caused by this bacteria in terms of their by toxins which is produced by the staphylococcus. And most of the common infections, they occurs after wounds, after operations, and this, if operative measures they are not good. Just a second. Okay, so this is another bacterial infection. Now in medical microbiology paper, the first unit, it totally contains the infections which are caused by the bacteria. Now up to now we have seen two diseases which is caused by the bacteria. First disease which we have seen is the cholera disease, and the second one is the disease which is caused by the Salmonella typhi. That disease we call it as a typhoid disease. And in that unit, first unit, there is another disease which is caused by the staphylococcus, and this staphylococcus infection is because of a group of bacteria which belongs to a staphylococcus. Now here we will begin this disease, staphylococcus infections, or it may be also known by another name, pyogenic infections. So this staphylococci, it is a group of genus which mostly contains gram-positive cocci. And the characteristic of this bacteria is that this bacterial cocci, they appear like grape-like clusters in morphology. When we observe this bacteria under the microscope, then we are able to observe this bacteria as a grape-like clusters. द्राक्ष जसे असतात, द्राक्षाचा घड जसा असतो तशा पद्धतीने तुम्हाला हे बॅक्टेरियाची मॉर्फोलॉजी तुम्हाला मायक्रोस्कोपच्या खाली पाहायला भेटेल. Now this is one of the uncommon character which is shown by this bacteria, and it is mostly able to cause some local suppurative lesions in human beings. We can say that pimples which occurs on the surface on the skins, on the face, these are caused because of this staphylococci. Now this bacteria, it also has the ability to resist the antibiotic penicillin and other kinds of antibiotics. But because of this is now one of the most dangerous pathogen, because this pathogen get resistant to various kinds of antibiotics. So because of that, if some operative measures has been carried out, and after that this bacteria is able to grow in that operative lesions, then it will cause serious problems. So this staphylococci bacteria were firstly isolated or observed by scientist von Recklinghausen. This is the scientist who observed this bacteria from the human pyogenic lesions. And here then the Pasteur obtained the liquid culture of this cocci from the pus and produced abscesses by inoculating this culture into the rabbit. Later on, the Pasteur isolated this bacteria in liquid culture and inoculated that in rabbit in order to observe what kind of effect it will have on that rabbit. So in that rabbit there will be development of abscesses. Then later on, Sir Alexander Houston, a Scottish surgeon, who established a causative role of this coccus bacteria in abscesses and other suppurative lesions. Okay, then another scientist who is the surgeon, who established the role of this staphylococcus bacteria in causing abscesses and suppurative lesions after the operations. Now, he also gave the name Staphylococcus to this group of bacteria because it is the staphyli, it is the word which is derived from the Greek, meaning bunches of grapes. Okay, so the Staphylococcus is this word. It is derived from the staphyli in Greek, which has a meaning that bunches of grapes. And now, due to the typical occurrence of this cocci in grape-like clusters, this cluster-like bacteria, they are observed in the pus of that wounds or post-operative measures. And here, this bacteria, when we observe under the microscope, same type of morphology, it is shown by this group of bacteria. The appearance of grape-like clusters in morphology. Now, this Staphylococcus, they are grouped in two different groups. That is Staphylococcus aureus, which is earlier known as Staphylococcus pyogenes, containing a group of bacteria which is showing catalase test positive, fermenting mannitol, and usually pathogenic. Okay, and the another group of bacteria, that is Staphylococcus epidermis, which is formerly known as Staphylococcus albus, containing the group of bacteria, group of strains which are coagulase negative, mannitol non-fermenting, and usually non-pathogenic. So we can compare these group of bacteria. Here, the Staphylococcus genus, it is grouped in two groups. Staphylococcus aureus, which is mostly involved in causing infections to human beings, and the Staphylococcus epidermis, it is not causing any kind of infections to human beings. Okay? We can easily differentiate these two group of bacteria by carrying out some certain test. Here you can easily observe that Staphylococcus aureus, it is catalase test positive. Here, catalase test positive is there. This bacteria can ferment the mannitol, and usually these pathogens, they are pathogenic. The Staphylococcus epidermis, this is another group which is catalase negative, mannitol non-fermenting, and usually non-pathogenic strains. Now, this genus Staphylococcus has been classified into 24 different species, depending upon the chemical composition of their cell wall and other properties. Okay, now next characteristic of this, now the bacteria Staphylococcus aureus, this is the bacteria which is mostly involved in causing pyogenic lesions in wounds and post-operative measures. Now we have to see the etiology of this bacteria, this disease which is caused by this Staphylococcus aureus. Firstly, the morphology of this bacteria. These bacteria, they are spherically cocci, which is measured up approximately 1 micrometer in diameter, and the morphology which these bacteria shows the gram positive cocci, which are arranged in a grape-like clusters, and this cluster formation is due to the cell division occurring in 3 planes, with daughter cell tending to remain in proximity. Means after cell division, these bacteria, they are not separated. So because of that, these bacteria remain attached and they form like clusters, and these clusters, they look like the grape-like clusters. Now they may also present singly, in pairs, or in short chains of 3 or 4 cells, but especially when examined from the liquid culture, long chains, this bacteria, they may present singly or in pairs, in short chains of 3 or 4 when we observe this culture which is taken from the liquid. Long chains of this bacteria, they are not observed. This group of this Staphylococcus aureus, it is non-motile, non-spore forming. This Staphylococcus aureus, this bacteria, it is non-motile, means they are not able to move, showing any kind of movement, and they are also non-spore forming. and not forming any kind of spores. Now, this few strains of this group of bacteria, they are able to produce microscopically visible capsule, and particularly this capsule is shown in young cultures. They are apparently non-capsulated strains and have small amount of capsular material on their surface. Oh, okay. Basically, they are able to, when these bacteria they are young, at that time, it are able to show the microscopically visible capsule. But later on, these bacteria they become non-capsulated. Now, these bacteria they can be easily stained with aniline dyes, and they are uniformly gram-positive. Now, under the influence of penicillin and certain chemicals, they may change to L form of bacteria. Here you can observe this is the Staphylococcus aureus, which are arranged in clusters, which look like a grape of clusters. Same gram staining is performed. You can observe this bacteria. When we observe this bacterial culture, which is taken from the liquid and observed under the microscope by performing gram staining, you are able to observe some bacteria they may be present singly, some may be present diploid, triploid, or tetraploid, and mostly they are observed in grapes-like structures. Now, next point in this is the cultural characteristic of this bacteria. This bacteria can easily grow on ordinary medium within a temperature range 10 to 42 degrees Celsius temperature, but the optimum temperature which is required for the growth of this bacteria, it is 37. And this bacteria can grow in a pH range between 7.4 to 7.6. Okay, so this bacteria can be easily grown in ordinary medium, or we can say that ordinary medium is particularly we use in laboratories. It is a nutrient agar. On nutrient agar, this bacteria can grow very well. And this bacteria, they show two kinds of aerobic and facultative anaerobic type of nature. Means they can grow in the presence of oxygen as well as in the absence of oxygen. So when we grow this bacteria on the nutrient agar medium, and after 24 hours we are able to observe the colonies which are large in size, having size 2 to 4 mm in diameter. These colonies they are circular, convex, smooth nature, shiny, opaque, and easily emulsifiable. Means we can easily take that colony and mix it with saline. This colony can be easily get mixed with that saline drop. Now most of the strains of this group, this bacteria, it produces golden yellow pigment, though some may be producing white, orange, or yellow. Here Staphylococcus epidermis, this bacteria mostly produces white type of colony color, while the Staphylococcus aureus bacteria, it mostly produces golden yellow colored colonies. And this pigment which is produced by this group of bacteria, it is not able to diffuse into the medium. Now pigment production occurs optimally at 22 degrees Celsius temperature and only in aerobic condition. In anaerobic condition, this bacteria it is not able to produce any kind of pigment. Now pigment production, it can be enhanced by adding 1% of glycerol monoacetate or milk into the medium, and this pigment which is produced by this bacteria, it is nothing but the lipoprotein which is allied to carotene. So this is the cultural characteristic of this bacteria. You can observe this is the growth of this Staphylococcus aureus bacteria on the nutrient agar plate. Here you can see the colonies which are large in size, which are circular, then showing convex nature, shiny yellow golden color, pigment produced by this bacteria. You can easily differentiate this from the epidermis colonies. So this is the colony of Staphylococcus aureus bacteria on the nutrient agar plate. Now when we grow this bacteria on nutrient agar slope, they form a confluent growth which represents oil paint appearance. Okay, if we paint our home with oil paint, then this home walls they become very shiny, Like that, when we grow this bacteria on nutrient agar slope, this bacteria produces oil paint like nature, which is showing very shiny color. Now, when we grow this bacteria on blood agar, they produces same type of colonies which are produced on nutrient agar, and the most of the strains they are hemolytic, means they can grow easily presence of in the blood on the blood agar. And when we specifically when we incubated this bacteria under 20 to 25% of carbon dioxide, and this group of this Staphylococcus, it shows beta type of hemolysis, and hemolysis is most commonly shown by this bacteria on rabbit or sheep blood, but beta type of hemolysis is not shown on the horse blood agar. This bacteria can grow on the McConkey's agar, which is a selective medium for E. coli. Here, it produces small colonies that are pink in color due to the fermentation of lactose. This bacteria can be grown in liquid medium where it forms uniform turbidity is there. Now, the various selective mediums are available for the isolation of this bacteria from, and these selective mediums, they favors only this Staphylococcus aureus bacteria to grow there, and then other kinds of bacteria they cannot grow on in these selective mediums. Now the selective mediums mostly includes 8 to 10% of sodium chloride. Now that particular medium which are used, you use this much amount of salt. They use the mannitol salt agar, salt milk agar, salt broth. Then another medium is there, lithium chloride and telluride medium. That medium is known as a Nudam's medium. And then another medium which contains antibiotic polymyxin. So these are the selective mediums which selectively allow the growth of only Staphylococcus aureus, not any kind of bacteria. Now for primary isolation, sheep blood agar, it is recommended. If we have to isolate this bacteria from any source, then sheep blood agar, it is very well medium for the isolation purpose of this bacteria. Now human blood, it is not used for the that is blood for hemolysis observation, because it contains antibodies and other inhibitors which inhibits the growth of this bacteria. So that's why it is that blood agar, it is not prepared from the human blood. So sheep or rabbit blood is used for the blood agar preparation, which shows the hemolysis. Here, this is the growth of Staphylococcus aureus on the blood agar. You can able to observe this is a colony which is showing the clearance zone around the colony, which is because of the lysis of the blood cells which are present nearer to this colony. So it is a hemolysis, and this type of hemolysis clear zone around the colony, that this type of hemolysis, it is known as a beta type of hemolysis. Here, this is the salt agar, salt milk agar, where this bacteria can show the clearance zones around the colonies. Now this is the McConkey's agar plates, where this bacteria shows small pink colored colonies on the McConkey's agar. You can easily differentiate these colonies, and these colonies, they are pink due to the fermentation of lactose sugar by this bacteria. Now this is another medium, selective medium, that is mannitol salt agar. You can easily differentiate. Here is the Staphylococcus epidermis colonies. These are the colonies of Staphylococcus epidermis, while these are the colonies of Staphylococcus aureus, which are showing yellowish pigment on the mannitol salt agar. Okay, so we can easily differentiate between these. So this Staphylococcus aureus, Staphylococcus epidermis, it is not able to ferment the mannitol on this medium. So because of that, there is no yellow kind of pigment is observed on around the colony. While in this Staphylococcus aureus, this bacteria is capable of fermenting the mannitol sugar. So that's why the colonies which are produced by this bacteria, they are of yellow kind, and the medium also becomes yellow, because change in the pH changes the color of the medium. Originally the color of the medium is this type. Okay, so it is a selective medium. Mannitol salt agar, it is a selective medium for the isolation of Staphylococcus species. Now this bacteria, now another characteristic in that etiology of this group of bacteria, it is the biochemical characteristics. Now this bacteria able to ferment various sugars, only producing acid, not the gas. Okay, this bacteria can utilize the sugar and produces acid, but this bacteria is not able to produce the gas in them. Sugar fermentation test, it is not diagnostic for this group of bacteria, but the mannitol fermentation test, it is diagnostic test for this group of bacteria, because this mannitol, this can be fermented by the Staphylococcus aureus in the absence of oxygen, but other bacteria, they are not able to ferment this sugar in the absence of oxygen. This Staphylococcus aureus, it is catalase test positive. This bacteria is able to produce urease enzyme and hydrolyze the urea. This bacteria is capable of reducing nitrates to nitrite. This bacteria is able to liquefy gelatin. This bacteria is able to show MR test and VP test positive, but indole test negative. Now this bacteria also able to carry out the lipolytic activity. When we grow this bacteria on a medium containing egg yolk, it produces dense opaque colonies. Okay. Now another test is also useful for the identification purpose of this group of bacteria. That test is we called as a production of phosphatase. And this test can be demonstrated by growing this bacteria on nutrient agar, which contains phenolphthalein diphosphate. When such culture is exposed to the vapors of ammonia, the colonies shows bright pink color due to the presence of free phenolphthalein. So this bacteria is able to produce the phosphatase enzyme, and because of the production of this phosphatase enzyme, it split this phenolphthalein diphosphate, and that phenolphthalein, which is released when it comes in contact with vapors of ammonia, it shows bright pink color. It is another test which is used for the demonstration purposes. Now, this bacteria, Staphylococcus aureus, we can easily differentiate this bacteria from other Staphylococcus epidermis. Now, by using this phosphatase test. Here, the Staphylococcus aureus, this bacteria is capable of producing this phosphatase enzyme. And when this Staphylococcus aureus colony is exposed to the vapors of ammonia, it is capable of showing the bright pink color. While the Staphylococcus epidermis colonies, they are not able to this group of bacteria, it is not Staphylococcus epidermis. This bacteria, it is not able to produce the phosphatase enzyme, and this because of the absence of this phosphatase enzyme, it is not able to use the phenolphthalein diphosphate. So phenolphthalein is not released, and when we take the ammonium vapors near to the colony of the Staphylococcus epidermis, there will be no pink color observed, bright pink color observed. Now, Staphylococcus aureus, which shows few characteristics which is not shown by other bacteria, this group of bacteria. First character, Staphylococcus aureus, it is coagulase test positive, means it can show the coagulation of. Then this bacteria, it is greater biochemical activity, ferment mannitol. This group of bacteria able to ferment, the Staphylococcus aureus is able to ferment the mannitol. This group of bacteria, this Staphylococcus aureus, it produces beta type of hemolysis on blood agar. It produces golden yellow pigment. It liquefies gelatin. It produces phosphatase enzyme. And this bacteria, when we grow in a medium which contains potassium tellurate, it reduces tellurate the tellurate and produces black colored colonies. Now this bacteria is also able to produce thermostable nuclease, which can be demonstrated by the ability of this boiled culture to degrade the DNA in the agar diffusion test. So these are the characteristics which are shown by this Staphylococcus aureus state. Okay, so we can easily differentiate the Staphylococcus bacteria from Staphylococcus aureus bacteria from other. When these tests they are shown positive by a bacteria, then we can say that bacteria which we have isolated, it is the Staphylococcus aureus strains. Here, this bacteria, Staphylococcus aureus, it is catalase positive, coagulase positive, oxidase negative. It ferments glucose, lactose, maltose, sucrose, mannitol, and produce acid, but no gas is there. It is indole test negative, MR test positive, UP test negative, positive, gelatin liquefaction test positive, phosphatase test positive. This is the catalase test. This is the catalase test negative. This Staphylococcus aureus shows the catalase test positive. This is the indole test. If this kind of ring is observed here, then it is a positive. But this test, indole test, it is a negative test for the Staphylococcus aureus. Here, this is the MR test, which is shown positive here. This is the VB test, which is shown positive. This is the normal medium. After utilization of production of urease enzyme, the slope becomes pink color. So this is urease test positive. This is the mannitol fermentation test, which is shown positive here. Now this is the phenolphthalein phosphatase agar medium, where this bacteria, after exposure to the ammonia, shows the pink colored colonies, bright pink colored colonies. This is the slide coagulase test, which is performed on the side. If there is a result, when there is a coagulase enzyme is there, then it causes the coagulation of this serum sample. Here you can observe this is the coagulation test, which is produced, this bacteria producing coagulase enzyme. So because of that, the serum sample get coagulated. This is the gelatin test, which is positive. It shows liquefaction of gelatin like this. If this gelatinase enzyme is not produced by this bacteria, then the slope, the gelatin stab, it remains as it is. There will be no liquefaction. But Staphylococcus aureus, this bacteria is able to produce the gelatinase enzyme, so it can liquefy the gelatin. Now, another characteristic in that etiology, the resistance characteristics. Now, this bacteria, it is more resistant among the known spore forming bacteria, other non-spore forming bacteria. Okay, this bacteria is not spore former. So among this non-spore former group, this bacteria is highly resistant to the various conditions. This bacteria can remain on dried thread for near about 3 to 6 months. This bacteria have been isolated from dry pus after 2 to 3 months. This bacteria can withstand 60 degrees Celsius temperature for 30 minutes. So the thermal dead point of this bacteria is 62 degrees Celsius for 30 minutes. You can observe that at 60 degrees Celsius temperature, this bacteria is able to survive. While other bacteria, they can be easily killed at 55 degrees Celsius temperature. Now, some of the Staphylococci, they require 80 degrees Celsius temperature for 1 hour to be killed. Okay. Now, this bacteria, it is heat resistant and have ability to grow at a high temperature even at 45 degrees Celsius temperature. Look that. While the normal pathogenic bacteria, they are capable of growing at the optimum temperature of 37 degrees Celsius and highest 41 degrees Celsius. But this bacteria can grow at a high temperature up to even at 45 degrees Celsius temperature. Now this bacteria is able to grow at high salt concentration up to 10% NaCl concentration. This bacteria can be grown. But the other bacteria, like that of coliform group of bacteria, they are not able to grow at such a high salt concentration. Okay, so this bacteria is salt tolerant. Now even at 15% NaCl, this bacteria can easily grow. So that's why we are using high salts for the food preservations. This bacteria can resist 1% phenol for 15 minutes. This bacteria can be easily killed by using mercury perchloride solution, 1% mercury perchloride solution in 10 minutes. Many aniline dyes, they are strongly bactericidal, such as crystalloyl, being lethal at a concentration 1 in 500,000. Okay, and billion means 1 in 10 millions. Now, this bacteria can be easily inhibited by presence of fatty acid. Here, the fatty acid inhibits the growth of the Staphylococcus, and highly saturated fatty acid having the powerful action on the coagulase positive than the coagulase negative strains. Okay, so highly saturated fatty acid, they can easily kill the Staphylococcus aureus species. Now, the Staphylococcus aureus, it is uniformly resistant to the lysozyme, but some micrococci are sensitive to it. Okay, Staphylococcus, they are generally sensitive to lysostaphine, a mixture of enzyme which is produced by the certain group of Staphylococcus epidermis strains. This strain produces this lysostaphine. It is a mixture of enzyme, and this mixture of enzyme can easily destroy the Staphylococcus aureus strains. So this is all about the etiological characteristic of the Staphylococcus infection.